Okay, for the next sort of 25 minutes, I'm going to give you an update on some work that we've been doing, looking at the relationship between early season retention and yield for high yield in Bollgard 3. And start off by acknowledging large team of people that have been involved in this work. As you'll see a little bit further on when I talk about the work we've been doing, it's very labour intensive. So I've had great assistance from Lisa Bradburn, Sean Holman, Gal Spargo, Jacob Balder, and Jamie Hopkinson within my team, based out of Toowoomba, DAF, and also at Emerald. And also Alison Young and Timothy Green, New South Wales DPI, that enable us to run sites in the southern parts of New South Wales. So a little bit of background around retention. And it'd be fair to say what constitutes ideal retention is a bit of a vexed issue. It's vexed for consultants and it's also vexed for your clients in terms of what the optimum might be. And in preparing for this presentation, I thought, well, let's go back and have a look at some of the more recent CCA surveys and get a bit of a feel for what it is that people are doing with regard to retention. And looking at that CCA data, if you take the average of the responses from respondents over the last couple of years, those respondents would indicate that people would expect to see a decrease in yield potential once they saw retention fall below 62% by the stage that cotton got to at first flower. However, if you look at the range of responses received in those surveys, there were also people that were quite content to have retention fall as low as 25%, and there are other people that are having kittens once retention falls below 90%. So quite a large range around that average of 62%. If you then look at some of the follow-up questions around cost of control, about 50% of farms are spending between $11 to $50 per hectare on mirrored management, with a further 41% spending more than $51. So that does represent a reasonable spend on quite a number of farms. But again, quite a broad range of views in terms of what retention might be ideal. So I thought, well, what is the origin of retention? Right? Why have we been measuring retention all this time? And when you actually go back and go through all the cotton pest management guides and look at a bit of the history of how we came about to be measuring retention, it was a key metric that was developed for the management of Helicobacter back in conventional cotton days. And essentially what it was about was Heliothis was so difficult to control that researchers at the time came up with a way to balance the damage that Heliothis was causing in conventional cotton with potential yield loss, with our ability to actually continue to sustainably manage that pest and not make resistance worse. And quite rightly, they found that cotton plant had the good ability to compensate, and basically that fixation with retention management was very much driven around retention of early bolls, because back in the day with conventional cotton, it was really, really difficult to keep that later fruit later in the season. So early bolls were a huge driver for yield potential within conventional cotton, and hence a lot of work around retention for those early fruit. Keeping in mind that yields around that particular point in time, about 25 years ago, were much, much lower than what they were today. Thinking about Bollgard 3 and factors that might affect Bollgard 3, the first point I thought was worth making is cotton's eternal. Cotton plant, it wants to live forever. It's a perennial plant. And it's naturally evolved to lose fruit and then grow fruit later on as a survival mechanism to get over all sorts of stress events. And with Bollgard 3 and Helicobacter largely been taken out of the equation, sort of think about some of the things that are affecting retention on Bollgard 3 crops. You've certainly got things like the weather, cloud, heat, cold, insects certainly have an impact on retention, and things such as suboptimal soil moisture or other crop stresses can result in that plant shedding squares and very young bolls. And basically taking the view that it's going to live to fight another day, and I'll go and make babies a little bit later on in my life. Mirids are a primary pest. We certainly all know that they affect square retention, they affect young bolls, they can tip out plants, and as a result can most certainly affect retention. And I don't think there's any need to dispute mirror management. Photo down there is photographs of cotton in Northern Australia last year at a remote site that only got looked at every couple of weeks, and huge mirror damage within that particular field. So there's no issue with whether or not we may need to control mirrors on occasion. But when I talk to a number of consultants, again, there's sometimes difficulties in integrating, for example, what your beat sheet might be telling you, what the plant might be telling you. Sometimes you can have high mirrors and good retention, other times you can have lousy retention and only very few mirrors getting around. And sometimes it's somewhere in between. So integrating what your beat sheet's telling you and what the plant might be telling you is not always the same year in, year out. And I guess it sort of raises this question of, can we better optimise our mirror management in terms of the wear of the plant? So thinking about the plant, we're sort of trying to build a bit of a picture sort of for you around this. Where have the increased yields in Bollgard cotton come from? So if you look at CSIRO data around this that Warwick Stiller and his team have generated, one of the big things has been increased turnout. So effectively smaller seeds that we all know about that are been a lot more harder to establish over the last 10 years than the varieties that went before has been a big part of it. Clearly a reduction in helicoverpa damage is a big thing, and not basically the, I guess, ability that we don't have to control helicoverpa in the same way now has allowed us to keep a lot more bolls in the outer and later canopy. And that's essentially where these big yields have come from. And if you think of some of the CSV data that's presented year in, year out, and I'll have some that will be similar later on, that's been a big part of those yield increases as well. And to go hand in hand with that, we have varieties that are a bit more indeterminate, but I'd say that we've got better behaved indeterminate varieties with the 70 family that reigns supreme, and they're just a cut above well and truly the old 189s, 289s that were our indeterminate varieties back in the conventional days. And that freedom around helicoverpa management has really allowed people to focus on agronomic management. Again, sorry did a study on this, a really good data set to look at what's been the contribution of cultivar versus management versus the interaction between cultivar and management. You can see sort of the numbers there, but basically management is contributing close to half of that interaction when you think about it separately, and then the combined interaction between cultivar and management. And essentially, a lot of those revolve around improved irrigation and improved nutrition management. So, before we come back to a more basic question, and that is to ask, it might seem counterintuitive, but how does cotton plant make its yield? And we know sort of in our heads that 65% of the picked yield of that crop, so that's what ends up in that module at the end of the field and the figure goes through as grown after cutout. Part of that potential is set before cutout. And we all know this, but the challenge I'd sort of throw today is, do we really understand this and have we actually thought through what that means? If you think about maximum fruiting sites, they're essentially set at around the time of cutout. And basically the period you go through between cutout and when you have those early first opening bolls, the plant enters this balancing phase where every day the assimilar production of that canopy is being matched by bulbamine underneath. Those bolls underneath are competing for that assimilate, and the plant's ability to retain those later set fruits is very much competition between those earlier set bolls and the ability of that canopy to produce assimilate day in, day out. And this is balancing act that goes on. You have a couple of days of cloudy weather just after cutout, tend to see a lot of shedding go with that, so the plant goes, oh, can't hang on to all of this, I might throw some on the ground. And really, ultimately, that final yield potential is absolutely underpinned by that period between cutout and about when 10% of those early bolls are open off those and data with that before. And again, we kind of know this, but we often really sort of think these things through. And it's why very much we see with crops that if you get stressed just after that cutout period, whether it be a heat wave, whether it's water stress, cloudy weather, whatever it might be, you see these crops roll off fruit, particularly from the upper and outer canopy on the ground. And that potential is not limited by the number of fruit that was there, but its ability to hold that fruit at that growth stage. When that internal struggle is complete in the plant, we start getting towards maturity. Most crops will retain less than 50% of the available sites that were produced by that plant in that time. And so you'd argue that for a lot of irrigated crops, that yield potential is not really limited by insufficient boll numbers at cutout, but it's by the ability to carry as many of those bolls from cutout through to maturity. That's really underpinning these very high yields. And that, as managers, I'd argue that our objective should be really about focusing on maximising leaf function during that period. And we do that intuitively. A lot of us already know that by managing our irrigation and nutrition to optimise leaf performance, manage leaf temperature, and basically have that plant doing everything that it can to hang on to those fruit during that period. And obviously preventing insects from removing bolls that the plant is invested in during the peak. So thinking then a little bit about development and fruiting site production. And some of you might have seen these couple of slides before from earlier CCA meetings a few years ago. But just thinking about what a plant looks like at first square. So first square developmentally, you'll have 3 fruiting branches at best. This is the first square, and then you've got a square with a subtending length, and you'll have a maximum of about 4 to 5 sites. If you sort of walk that through and you think, okay, what's a plant likely to be able to produce by first flower? By first flower developmentally, because not every plant's going to develop like this, but if you think about the accumulation of day degrees and what that plant's achieved, you should have about 20 to 21 sites per plant. Certainly there's been enough day degrees to accumulate that many fruit. And again, 2 weeks, 15 days in the first flower, you're potentially up to about 37 sites that have been produced by that plant, assuming you're a reasonably vigorous crop and you're still travelling at, say, about 6 nodes above white flower. And you can see in the graph here that production of fruiting sites becomes somewhat exponential in pattern. And the reason for that is you're growing nodes vertically and also growing nodes horizontally with the extension of those fruiting branches. So if we sort of use that as a bit of a background for the research work that we've been doing, and one of the difficulties with murids is they're incredibly difficult insect to work with. So the approach that we've taken to better understand this relationship between retention and yield has been to inflict artificial damage at a number of sites. And we do that damage quite mechanically, so everyone gets armed with these little spring-loaded pliers, just basically give the square a pinch, 3 or 4 days later it falls off, a bit like a murid doing damage. Obviously not the same, but the closest thing that we were able to come up with. And what we've essentially done is we've come up, first year we tried a whole range of different treatments, and this year we've sort of narrowed it down a bit to where we have an undamaged control. We then have removed the squares from the first 5 fruiting branches. So everything that's on those fruiting branches comes off at that stage. To give you a rough idea, it's about 7 to 10 days out from first flower. So if you think about that graph there, we're sort of aiming for that zone there, taking everything off the plant. If you think about that diagram, you end up with this plant with a hole underneath it. We then had a second treatment where we did the same thing again, but took fruit off the next 5 fruiting branches, 6 to 10. In effect, sort of creating a bit of a hole further up in the canopy with some retention underneath it. What we're interested in there was whether those early holes would limit the ability of the plant to compensate further on, because there'd be some within canopy competition going on. And then last treatment, just to really kick the ball out of the park, we acted on plots of plants, those 2 damage events, one after the other consecutively. When we did the second damage event, we don't go back down to the first 5 fruiting branches, so they start to compensate, left them alone, and just re-damage the next 5 fruiting branches in the same manner that we did that second treatment. To give you an idea of the measurements that we took of this, we get really detailed measurements of fruit number, fruiting site production over time, the production of biomass, and then partitioning that biomass. How much of that biomass was going towards leaf production, stem production, bulb productions. Detailed assessments of crop maturity, a segmented pick on these plots at the end of the experiment to determine how compensation was achieved, bulb size, some of those factors, and then obviously did machine pick yield where we could. Sometimes it was hand picking depending on the location, and leaf quality. And the trial sites, this year we had 5 different sites from Emerald through to Witten in southern New South Wales. Those sites are just managed as per commercial fields. So we go into the corner of a commercial farm, map out these plots, chip out some alleyways, and that paddock gets essentially the same management that the rest of the paddock's getting. It's not getting anything in addition to what the rest of the field has, apart from during those early stages, we might do an additional murid spray, only because when you pull the squares off, I think I worked out this year about 2 kilometres a row, you don't want a bunch of murids coming in overnight and wrecking your treatments. But apart from that, same water, same nutrition, same pretty much everything else, time of year for foliation, etc. So we'll start firstly with leaf yield and quality. This is just the data from this year. We've got the same result last year. You basically see that there was no significant differences in yield between any of those treatments, even that most extreme one where we've stripped pretty much all the fruit off the first 10 fruiting branches. You can see a bit of bounce in the data, but essentially across all those sites, there's no significant difference. And then you argue that the replicate differences are as great as what the treatment differences are. The one on the end, we tried this also at a later planted skip row site at Dolby. We're interested to see sort of how different row spacings might interact with this, but again, got the same sort of result. No difference in quality at any of the sites, including in the far south, with one exception being slightly improved microneria at Emerald, but again within base grade, and all sites produce length within base grade. In terms of the fruiting dynamics, and this is where you start unpacking, well, what is going on with these plants. You can see this is just retention over time. Now this is total plant retention, so total number of sites, we take a metre of row out of each of the plots back to that, pull it apart. You can see that certainly where we've taken those squares off, it had a pretty big impact on retention. But basically, then when the crops are left to grow out, by the time you get the cut out, retention is pretty much evened up across all those treatments despite those setbacks. And the reason for that is where we've taken that damage off, plants are growing more fruiting sites. So we've got extension of those branches sideways, growing a bit more stem and leaf and new squares. and also some additional sites in the upper canopy. And effectively, you see the control there, but the other sites in that really extreme damage one, it's 25% more sites produced. But the interesting thing about it is those sites are all in place and have all been produced by cutout as per that control that was undamaged. And if you think about then fruit numbers, again, that cutout, we've got a lot more fruit that are all fruiting sites, squares and young bowls that have been produced where we've done damages, compensation. And that compensation, everything's in place by cutout. And this starts to explain why we're not seeing yield and maturity differences. And in fact, if conditions were really good, if you think about what we talked about before between cutout and early bowl opening, potentially yield increases, which is what we had at Gundawindi last year with some of the treatments. Weather was actually a bit better after cutout, and those treatments actually held a bit more fruit. In terms of biomass accumulation, as you'd expect, a bit more stem and leaf. That goes hand in hand with the production of those extra sites. Bowl accumulation, so you see the control there, quite a more rapid accumulation of bowl weight over time, but you can see how the other treatments, again, sort of by that first open bowl, between first open bowl and crop maturity, basically catch up. So they're a bit slower to get going, as you'd expect with the loss of those early bowls, they catch up. As all of those fruiting positions that are there, present, they cut out, fill out, and go forward. And you can see, as you'd also expect, slight increases in leaf area associated with the stem and leaf that's produced by the crop. This data is just an average across all of the sites, but pretty much the same pattern at each and every site. Now, probably a better representation of that is, I think this habit of taking pictures of biomasses as we partition them. So the first thing we do is pull the leaves off, so they go through a leaf area machine. And it's just interesting to throw them on the floor and take a picture. So you can see here the control, fairly typical for all these sets of photos. This is the Gundawindi side over at Newell's place. You can see there we've taken the bottom squares off, bit of a hole underneath. The next treatment, bit of a hole in what would be the middle later on by the time this is grown a bit more. And then what looks like fairly flogged out cotton. So you can see the damage we're doing is quite extreme, quite excessive, and far beyond what most people would be comfortable with. But for the sake of exercise, these treatments. If you then look at those same plots that cut out, you can see that, again, there's that hole at the bottom, there's a bit of a gap in the middle, and then these ones here. But basically, at that point in time, the number of fruit numbers that are there are pretty much the same, actually more in these treatments than what is in the control. So potential is potentially greater at that point in time. And then if you look at maturity, and this is kind of interesting, control as you'd expect. So we do these biomasses when about 50% of the bowls are open as per the control. Arbitrary maturity dates, so we can get this work done before the first foliations go out. But you can see in this really extreme one, very few open bowls, but if you look at the size of these top fruit and compare it with theirs, very, very similar. And to sort of put that into perspective, when we start looking at the maturity data that's collected from these crops, what you typically see, this is the control blue line, and it's just 3 sites as a comparison. But you can see how much damage treatment starts off really slow with bowl opening, but then it catches up towards the end. And these other 2 treatments where we've got more of a hole in the middle of the canopy very much do the same thing. If you like, what happens is they open slowly at the beginning, but because there's more bowls of a similar age further on, it just catches up. It's a bit like baking popcorn in the microwave, pops a bit slowly and away it goes. And again, maybe sort of pictures speaks 1,000 words, but this is Gundawindi, 4 days after the 3rd pass. This is the control, and you can see we're just waiting for those last bowls to open up. It's had a pick about 3 or 4 days later. But again, you look for argument's sake at this particular treatment where we put a hole in the middle of the canopy. You can see those last bowls are very, very similar. Might be in a practical sense 2 to 3 days behind, worst case scenario, but very similar. So you can sort of see practically how this is working its way through. Again, at Brookstead, another example, this is the control. So again, waiting for those last couple of green bowls that are through the canopy. This is after the 2nd pass. And again, this is that extreme treatment. So, this is what it looked like 3 weeks after you did the damage. And yet, look at the green bowls that are in there. A couple more, but again, extreme damage. It's sort of taking pretty much everything off those first 10 branches. And again, the yield is the same. And the maturity is very, very similar. And this goes a long way to explaining why the quality differences weren't there either. A little bit about segmented picking. A lot of you have seen this data before, but for those of you who aren't familiar with it, essentially what these graphs are depicting is, imagine the y-axis is the main stem, and then what we've got is different branch groups. And in this case, what we've done is we've segmented, picked the canopy, put all the vegetative branches together, and we've looked at the first 5 fruiting branches, the next 5, the next 5, and so forth. And it gives you a bit of a map of the canopy. So you see that's the number of bowls that were on a metre of veggie branches, and bowls that were on those first 5 fruiting branches here, the next 5, the next 5, and then basically any fruit that are in second or third position further out in the canopy. And if you start sort of looking at yield and the distribution of bowls in these treatments, and again, this is just an average across all of the sites except the skip row one at Dolby, that first position bowls were reduced quite markedly by a move of fruit. Didn't get rid of every single one, because again, we go in, we take an average point where we enact this damage, might be the odd plant that's behind developmentally, might have squeezed out, a position on the 4th or 5th fruiting branch after we did the damage, but you can certainly see the impact of what we did relative to the control down here. What we're seeing though is you get very immediate compensation. So you have a rapid increase in the number of fruit held in that first position, bowl position in the branches immediately above where we did the damage, and clearly an increased number of P2 and P3 fruit on the branches in which the damage occurred. So that compensation is very rapid, it's very immediate, it's not all up the top of the plant, it occurs throughout the canopy and it occurs more or less adjacent to where that fruit was lost. And that goes a long way to explaining why we're not seeing loss of maturity and necessarily yield impacts. In terms of bowl size, very little difference when you average it across the entire canopy, but what we consistently see is the bowl beside where you've lost a square will be a bigger bowl. So again, compensation is happening very locally within the canopy, but when you average out bowl size across the canopy, more or less the same. And just another way of depicting it, this graph here just sort of shows where bowls were lost from the canopy due to the 3 treatments relative to the control. And you can see where the extra bowls were retained as compensation within the canopy. Again, it occurs throughout the canopy. So what does it all mean? I guess when you start thinking about Bollgard 3 in our high yielding systems, basically these crops have been able to compensate for those loss of early positions. And if you think about how yields achieved in Bollgard 3, we are not as dependent on those early positions as what we were in the days of conventional farming. The other things to sort of really think about is 75% of the positions that we rely on for that yield potential aren't even produced at the stage of first flower. So 25% of what's going to be there is there at first flower, but the rest is still in front of us and yet to materialise. Compensation, as I just said before, is pretty much occurring immediately. It occurs on the same branches where you've lost fruit, occurring immediately above those branches. And if you start sort of doing the mental thinking through what this actually means, if a crop's got time to produce 12 to 15 fruiting branches, and I would argue that's a prerequisite for a high yield irrigated cotton crop, then you've got ample time from a data grid perspective to compensate from the loss of fruit from those first 5 fruiting branches. Talked about this with a number of people from the south, and our season's too long. It's like, how many fruiting branches do you go? Oh, at least 12 to 15. If we grow 12 to 15 branches this way, you can well and truly get enough compensation that way as well as done. And that's why we didn't see any differences at Whitten, or basically the crop performed the same at Whitten as what it did at Amon. And so the basic takeout is that you can have substantial square loss before flowering, and without it really interfering with core metrics of how that crop's going to grow. However, you could also look at the data that I presented today and play devil's advocate. You could say that the data showed that there's also no negative impact from the controls of high retention. And therefore, managing for high retention is a justifiable sort of way to go. And at the end of the day, that makes sense to me. Like, why would a sensible person take the risk with playing around with retention early season, particularly when you can chuck it to methylate or fipronil in with an over-the-top glyphosate that's going to go out anyway? And as someone I ran this by said to me, if I came onto the farm and glyphosate was going out Monday and I didn't put anything in with it, and I came back to that farm and do the next check on Thursday and there's mirrors and I could do a separate mirror spray, I wouldn't get asked back to that farm again. So we kind of get the reason why we do what we do. And the other thing is, most people are going to be way outside of their comfort zone with cotton, and anything like this or this, and we won't even contemplate our extreme treatment, which is kind of risky fix, but trying to make a fairly strong point. And comment I'd make, no one's probably ever really been sprayed sacked for putting on extra spray. But I think there's a bigger picture, and Paul's touched on it in his presentation this morning. I think sort of some of the information that CRDC talked about and Susan put together. have sort of suggested that, you know, there's a bigger picture going on, and there's really good evidence that some of our early season insecticide use early on in the season is certainly flaring some of that things like silverleaf whitefly. It doesn't happen every year, and I have this conversation with consultants again and again, where like I've got two people, neighbouring farms. One guy likes to go soft, the other guy, you know, likes to go the low road and go anything that's cheap. And they both ended up doing a whitefly spray anyway, so that guy spent $60 more per hectare than that guy. Yeah, that happens. But if you look at it on balance, season over season, there's really good evidence. Richard's been looking at silverleaf whitefly population dynamics in commercial farms now for about 3 or 4 years. Very clearly in those high whitefly years where you see you can correlate a more rapid increase in silverleaf whitefly and the need to spray more than once in fields that have been disrupted. So it doesn't happen every year, but it certainly is a thing. And if you think about it, like particularly when we had those hot dry finishes a couple of years ago, it was quite stressful for a lot of people trying to prevent honeydew when we got pinged overseas with honeydew picked up in some of our exported mint. And then you think about those very bottom bowls, if you've gone all the trouble of retaining those first couple of bowls, they're the ones that are open for the longest, sitting there catching all that honeydew for the longest period of time. Just for something else to sort of keep in mind. Think about those maturity opening grafts, are exposed for a long time. So again, something to sort of keep in the mix. This is not just about virid management, it's about the management of these other pests in the system. Think about those environmental toxic loads that within the CRDC presentation, that fipronil usage, the methylate usage early season, just as an add-in with glyphosate is being reported, it's being picked up. So that's part of those metrics again, it's part of that bigger picture. Is it very easy to justify when you think about the slide before, but there's other aspects to all of this, there's other moving parts. And I guess the question would be, if and when we lose those products, in the case of fipronil and methylate, we suggest that probably the next 5 years we'll see them gone, would you spend $20 to $30 on a transform on a plant that's per square and got 3 or 4 breeding branches in light of that sort of data? So I think we've got an opportunity, you know, we've got a few years before we need to start thinking about making these changes, but it's sort of quite thought-provoking how this plant wants to respond. So if I was to leave you with a take-home message for today, I think we have a real opportunity to start the process of recalibrating our thinking around what acceptable levels of square loss might look like on Bollgard 3 crops, and start experimenting with that, getting a feel for it, becoming more comfortable with it before IPM or legislative changes or whatever it might be starts making throwing in a $5 product with an over-the-top glyphosate a lot less attractive. I think also there's a real opportunity to start developing a better and deeper understanding of how the cotton plant grows, how it produces its yield, what are some of those key stages, and I think that information we could use to better optimise some of our management. And I guess to that end, how we're approaching some of the extension of this work over the next season or two is rather than having an argument about whether or not we need to manage mirrors. I think that's reasonably counterproductive. Cotton Info are going to work with us, and we're going to run demonstration sites in each valley throughout the industry this year and hopefully the year after, for the purpose of demonstrating how the crop grows. Have some exercises where people go in and do different things, might be different damage, might be tagging files. I think we're going to go through the maturity, but basically have a really in-depth conversation about how that plant grows, how it develops, and so people can become comfortable with what this plant can actually do and what the limitations and boundaries might be. And then perhaps with that level of confidence, we can start being a little bit more prepared to push the window with some of our management of insects, pests, early in the season. And we certainly really look forward to welcoming your involvement in those exercises that they'll be in each valley this season. So keep an eye out for that. So just like to sort of finish up just acknowledging a few people. Certainly like to acknowledge our trial site hosts, Ken Giddings and Emerald, Matt McVeigh at Dolby, Paul and Georgia Krieg at Brookstead, Andrew and Rob Mule just here at Gundawindi, Rhys Thomason, Roger Collins and Tim Commons at Whitten. So again, happy for us to come into their field, cotton out, we need to make alleyways to get hot pickers in and things like that, and let us do our thing within their paddocks. Dave Kelly's been a tremendous help. We've had a trial based here at Gundawindi a couple of years running now, working with Dave really well. Also Warwick Stiller and his team at CSI Breeding. I tend to follow Warwick around wherever I can. He's got this really fancy picker, and it's like a 2-day job for us. He does in about 20 minutes, so wherever I can piggyback Warwick, I do. The study has been funded by the CRDC Project 1902. Thank you.
Great presentation, Paul. Very thought-provoking, and there's a massive amount of work that goes into those trials, so very well presented. Got enough time for one quick question if someone's got one? We've got a question on Zoom as well. You, Brian?
Very good presentation, Paul. There are parallels among me and speak about when first grade. So, yeah, before I retire, we should catch up.
Sounds like a day. So the questions we've got on Zoom, was there any change in yield across the different varieties and how they responded to damage at different times, like 746 versus 748?
Yep. So just quickly,
Just so you're in the
this
camera.
one. Oh,
Yep,
yep.
so you're in the camera.
Oh, there we go. Okay. So just quickly, so this last year was 746. It performed just well. This year, like I said, we just go into fields, we haven't got variety comparisons. But 4 of the sites were 748, the site in the south was 746. So it's now across 7 sites, we've got 3 746 and the rest is 748, and we're seeing the same thing.
So the next question was, if there was adverse environmental conditions in the period following cutout, which bulb position would you suggest would be at risk most of shedding?
So yeah, look, that's certainly a factor. If you had really late stress, you're reliant on a high proportion of young fruit at that stage in the canopy, you could come slightly unstuck and lose a little bit more of that potential compared to a crop that had locked away more of that yield earlier on. But again, it can also very much go the other way. In that last year, we sort of had a heat wave event down here at Young and Windy, and the crops that had bulbs at a slightly younger stage actually handled that a lot better and yielded about 1.5 more than what the controls did in a number of those treatments. So again, a little bit luck of the draw. It goes to show how important doing what we can as managers to minimise that stress, particularly between cutout and early open bulbs. But also the other thing to keep in mind is that crop also has slightly bigger leaf area and a little bit better capacity to handle some of those stresses being slightly bigger. So there's lots of swings and roundabouts there, but I think it's telling that from emerald to Whitman, you look at the biomass data and effectively all of those crops have pretty much come in with an identical number of bulbs, an identical yield, and that very much says that yield potential is capped by the management of that paddock and the environmental interaction that's taking place. It's had very little to do with the retention of early fruit.
One more question on Zoom. Does this mean that mirror thresholds potentially can be reviewed again?
Yeah, look, we're hoping, one of the things I've got to mention is we're repeating this work for at least 2 more seasons. Because one of the key things, obviously, is to get seasonal variability and also just to gain confidence that what we've seen over the last 2 seasons is sort of more or less holding true. We're then hoping to be able to make some fairly firm recommendations, not so much around specific mirrored numbers, but certainly around the plant and how we should be managing around retention. And sort of treating it from the point of view of looking at it from a crop retention perspective. So the way I tend to think about cotton is that plant is the integrator of everything that's going on in the paddock, whether it's the mirrors doing damage, whether it's the weather, and some of those different things. And yeah, we're sort of hoping that we'll be able to come out with some sort of, I guess, dynamic retention model for cotton going forward that'll be compatible with high yields and also some of the IPM challenges that